Adrenergic transmission, also known as noradrenergic transmission, is a crucial function of the sympathetic division of the autonomic nervous system. The primary endogenous catecholamines involved are noradrenaline, adrenaline, and dopamine. Noradrenaline acts as a neurotransmitter at postganglionic sympathetic sites, except in sweat glands, hair follicles, and some vasodilator fibers. It also functions in specific brain areas. Adrenaline is secreted by the adrenal medulla and may act as a neurotransmitter in the brain. Dopamine is a major neurotransmitter in the basal ganglia, limbic system, chemoreceptor trigger zone, anterior pituitary, and to a limited extent in the periphery. Catecholamines are synthesized from the amino acid phenylalanine through a series of steps. The process begins with tyrosine hydroxylase, a specific and rate-limiting enzyme in catecholamine synthesis. This enzyme is inhibited by a methyl P-tyrosine, which depletes catecholamines and is used in managing pheochromocytoma. Other enzymes involved include dopa decarboxylase, which can also produce serotonin from 5-hydroxytryptophan and a-methyldopamine from a-methyl-dopa. Noradrenaline synthesis occurs in all adrenergic neurons, while adrenaline synthesis occurs only in adrenal medullary cells, requiring high glucocorticoid levels to induce the methylating enzyme. Noradrenaline is stored in synaptic vesicles or granules within the adrenergic nerve terminal. Dopamine is taken up into vesicles, where it is converted to noradrenaline. Noradrenaline is stored as a complex with adenosine triphosphate, ATP, in a 4:1 ratio adsorbed on chromogranin. In the adrenal medulla, noradrenaline diffuses into the cytoplasm, is methylated to form adrenaline, and is taken up by separate granules. This storage mechanism ensures that catecholamines are readily available for release when needed. Catecholamines are released by exocytosis, which includes noradrenaline or adrenaline, ATP, dopamine B-hydroxylase, and chromogranin. Presynaptic receptors, primarily alpha-2 receptors, modulate this release. Autoreceptors of other cotransmitters, such as Y2 of neuropeptide Y and P1 of ATP, also inhibit release. Indirect sympathomimetics, like tyramine, induce noradrenaline release by displacing it from nerve endings without exocytosis. This complex release mechanism ensures precise control over catecholamine levels in the synaptic cleft. Axonal uptake, also known as uptake 1, involves the recapture of noradrenaline by an active amine pump at the neuronal membrane. This mechanism terminates noradrenaline's action, with 75 to 90% of released noradrenaline retaken into the neuron. This process is inhibited by drugs like cocaine and desipramine. Vesicular uptake involves the VMAT2 transporter, which moves catecholamines from the cytoplasm to the vesicle interior, maintaining neuronal noradrenaline content. This process is inhibited by reserpine, leading to catecholamine depletion. Extraneuronal uptake, or uptake 2, is conducted by ENT or OCT3, capturing circulating adrenaline, but is of minor physiological importance. Catecholamines are metabolized by two primary enzymes, monoamine oxidase, MAO, and catechol-O-methyltransferase, COMT. MAO acts on noradrenaline in the cytoplasm, and those taken up by axonal transport. COMT acts on catecholamines that have diffused into circulation, primarily in the liver. The end products of catecholamine metabolism, such as vanillyl mandelic acid, VMA, are excreted in urine, providing a means to measure catecholamine turnover in the body. Adrenergic receptors are G-protein-coupled receptors located on cell membranes. These receptors influence intracellular second messengers like cyclic adenosine, monophosphate, CAMP, or inositol, trisphosphate, diacylglycerol, IP3/DAG. Activation of these receptors can also affect ion channels and prostaglandin production. This intricate signaling system allows for diverse physiological responses to catecholamines, ensuring that the body can adapt to various stimuli and maintain homeostasis.